Good evening, students. Yesterday, we discussed up to incomplete dominance in principles and variations. Today, I will continue the chapter with the exception of Mendel's law. I hope yesterday all are recalled well, because recalling, listening, recalling, and taking hints, which is most important than the study. Listening, taking hints, and recalling, which is most important than the study. You must follow that thing. I hope our students will be follow those things. So today's topic, I will start with the exception of Mendelism. Mendel's statement is one gene is responsible for determining the one character, and in a pair of genes in a gamete, one is dominant, another one is a recessive. This is a Mendel statement. This is a Mendel's law. But in some cases, one gene can control the many character. One gene can control the many character. Sometimes both character will be expressed. Sometimes new character will be expressed instead of that parents. Clear? So those collectively called, those are the collectively called, that things are called as a exception of Mendelism. Those things are called as a exception of Mendelism. So what are the exception cases? Means 4 exception case will be there. What are they? Means the first one is a incomplete dominance, second one is a co-dominance, third one is a multiple allele, fourth one is a pleiotrophic genes. The first one is a incomplete dominance. Yesterday we discussed. I mean that new character will be a formed, mixed character, new character will be formed. That is called as a incomplete dominance. The second one is a co-dominance. Second one is a co-dominance. So what's the meaning of second exception case is a co-dominance? What is the meaning of co-dominant one? Co means along with. Go means along with. Dominant means the particular character will be expressing. That is called as a dominant. So particular character will be expressing. That is called as a co-dominant. So one character will be expressed along with the another one character. That is called as a co-dominance. So simply we can say both parental character will be expressed equally. Both parental character will be expressed equally. That is called as a co-dominant one. Here in co-dominant, no dominant, no recessive relationship here, because there is no dominant, there is no recessive gametes, because that both character will be expressed simultaneously. That's why which is known as a co-dominant one. For example, so many examples will be there. First example is a ABO blood grouping. ABO blood grouping. That ABO blood grouping is discovered by Lance Steiner. That is an important neat question mark. Kindly note it. Lance Steiner discovered the ABO blood grouping. In human, that the gene is responsible for determining the human blood group. The gene I is responsible for determining the ABO blood group in human. Mostly in different organisms follows the different kinds of blood groups. For example, in humans, we are following the ABO blood groups. Humans, we are following the ABO blood groups, that each blood group having the antigens and different antibodies. For example, blood group A consists of the antigen A and the antibody B, like that. It shows us some variations. We will discuss one by one. ABA blood group is an example for the co-dominant one. First one is a blood group. A blood group. I means here I means a gene responsible for determining the blood group. A means a antigen. A means a antigen. So that RBC of plasma membrane can able to produce the A antigens in A blood group. Similarly, B blood group. In B blood group, RBC of plasma membrane can able to produce the B antigens on the surface of the plasma membrane of RBC. So A blood group consists of the A antigens. B blood group containing a B antigens on the surface of RBG or BC, red blood cells. While we are crossing the A and B, while we are crossing the A and B, at that time we can get it both A antigen and B antigen. Clear? When we are crossing the blood group A and B, at that time progeny, that offspring may consist of the A antigen and B antigen. That blood groups are a AB blood group. So if the one person is having the A blood group, another one person, the woman is having the B blood group, means when we are crossing, at that time that progeny is a progeny blood group is a AB, it containing both A antigen and B antigens. That both character will be equally expressed. A antigen and B antigen, A B blood group both will be expressed equally, which is called as a codominant. Codominant. Here A and B is a dominant, O is a recessive one, because O blood group does not having any antigens, but it having the antibodies. A blood group and B blood group having the antigen. O blood group does not having any antigens. That's why O is a recessive one. Another one important thing is when O is combined with A or B antigen, at that time O does not express it. That is important, Mainma. When O is combined with A or B, at that time it does not express it, because which is O is a recessive one. Here we are going to we are taking the two parents. First one is a parental 1, second one is a parental 2. Listen the table. First one is a parental 1, second one is a parent 2, third is a gene type, fourth one is a offspring, progeny. The first parental is a parental 1. While we are crossing the antigen A, I mean A blood group and B blood group, at the time genotype will be a IA and A antigen. Genotype carries a both genotype, both are a antigen A. That blood group is a A blood group. Similarly, in second one, when we are crossing the B blood group, at that time which that both are parental 1 and parental 2, that both B blood group, parental 1 having the B blood group, which is crossed with the parental 2, also having the B blood group, that both are having the B antigens. So at that time genotype will be a B, IB and IB. At that time that progeny blood group is the B blood group. The third case, that parental 1 is a blood group, which is crossed with the B blood group. So at that time parental A having the parental 1 having the antigen A, parental 2 having the B antigens. So the genotypic one is I mentioned in green color theme. Genotype carries the A and B. So blood group is AB. Blood group is AB. Then fourth case, IA. Parental 1 is a blood group. Parental B is a O blood group. O blood group. At that time, genotype is A and O. Just now I told, while the O is combined with the A or B blood group, at that time O does not express it, because O is a recessive one. So our blood group will be a A. The fifth case, parental 1 is a B, belongs to B blood group. Parental 2 is a O blood group. Then the parental blood group B is crossed with the O blood group. At that time genotype will be B. Genotype will be IB and IO. So blood group is a B. Blood group is a B. So last case, in sixth case, that both parental 1 and parental 2 cross, the both parental group is a O. Both parental blood group is a O. So that O blood group will be crossed with the O blood group. At that time genotype will be IO and IO. Progeny blood group is a O. Progeny blood group is a O. Here we can check the phenotype. Phenotype means how we can calculate the phenotype means blood group ratio. That is a phenotype ratio. Phenotype. How many blood groups we are getting in codominance? Totally 4 kind of blood group. What are they? First one is a blood group. Second one is a B blood group. Third one is a AB blood group. Fourth blood group is a O. So we are getting a 4 kind of blood group. That is a phenotypic one. Next one is a genotypic. How many genotype we are getting? Totally 6. First one is a antigen. Second genotype is a B antigen. Third genotype is a AB antigen. Fourth genotype is a AO antigens. O is a recessive one. Similarly, fifth one is a BO antigen. O is a recessive one. The last one is the O antigens. So totally we are getting the 6 types of genotypes. So how many parents we are taking? Totally 3. How many parents we are taking? Totally 3. Parents means A is a one parent. A is a one parent. I mean blood group A is one parent having the I A antigen. B blood group having the B antigens. The last one is a O blood group. So totally 3 parents we have. So what are the other experiments for codominance? Means the carrier of sickle cell anemia. Carrier of sickle cell anemia is another example of codominance. Sickle cell anemia carrier having the HPA and HPS genes. Another third example for codominance is animals of coat color. Animals of coat color. Animals of coat color. For example, when black colored cat is crossed with white color cat, when black color cat is crossed with white color cat, at that time progeny should be in progeny that both color will be expressing. For example, that progeny having the offspring having that black with white in color. So that is a third example. So when black color cat is crossed with white color cat, at that time progeny having the black and white coat. So there is a third example of a codominance. Third exception case is a multiple allele. Third exception case is a multiple allele. So what's the meaning of allele? Yesterday I gave definition. Simply we can say allele means a 2 alternative form of genes. Allele means a 2 alternative form of genes. That is called as allele. For example, caps T and small t. That is a 2 alternative form of genes. Caps T is a 1 alternative form. Small t is responsible for the trough. That is another one alternative form. The 2 alternative form of gene, which is called as a allele. If 3 or more than 3 or more than 3, if allele is having the 3 or more than 3, which is known as a multiple allele, Human blood group, because commonly we know very well, mostly the two things are present. For example, caps T and small t. In mental statement, 2 alleles will be presented to control the particular character. But some cases, 3 or more than 3 alleles will be involving. That is called as a multiple allele, involving to control the or to determine the particular character. That is called as a multiple allele. For example, human ABO blood group. Just now I told what are the alleles? A, B, O. Those are the 3 alleles. Those are the 3 alleles can control the human blood group. So A, B, O is a 3 alleles which is control the human blood group. Clear? The second example is skin color of human. Skin color of human, which is controlled by the 3 alleles, namely A, B, C. Namely A, B, C. So caps A, caps B, caps C, which is responsible for dominant one, which is responsible for gives a dark in color, which is a dominant one. Small letter A, small letter B, and small letter C, which is responsible for gives a light skin color. Light skin color. Caps ABC is responsible for dark in color. Small letter ABC is responsible for gives a light skin color. So we are going to cross the 2 caps letter ABC, dark color giving alleles, and a small small letters, I mean light colors giving color alleles will be crossing together. So at that time, that gamete will be segregated as usual. Capital letter ABC will be segregated from the parental one. Similarly, small letter ABC also segregated from the recessive parental one. The both will be combined together and forms a intermediate skin color. And forms a intermediate skin color. The progeny does not having the darkened color or a light skin color, having the intermediate. Intermediate means middle of the two color. That is called as a intermediate. Next one is a pleiotrophic genes. Next exception case is a pleiotrophic. What's the meaning of pleiotrophic? So that pleiotrophic gene is against the law of dominance. Against the law of dominance. Because one gene can control the one phenotypic traits. But in the case of pleiotrophic gene, a gene influences two or more unrelated phenotypic traits. A gene influences two or more unrelated phenotypic characters, which is called as a pleiotrophic genes. A single gene can able to determine the two or more unrelated. That function is that character is unrelated. It can able to control pleiotrophic genes. Examples: sickle cell anemia. First example is a sickle cell anemia. So when the person is suffered from the sickle cell anemia, sickle cell anemia causes of various reasons. Majorly, mutation is a major causing agent for a sickle cell anemia. The sickle cell anemia carriers having the HBS genes. HBS genes. That's that character can controlled by the sickle cell anemia character can controlled by the HBS genes. What will do the HBS genes means hemoglobin synthesis will be reduced up to 50%. Hemoglobin synthesis will be reduced up to 50%, because you know very well that what is the meaning of sickle cell anemia? Anemia, what is the meaning of anemia? Anemic means which is related to the blood, You know very well RBC, totally the two kinds of blood cells will be present. One is the WBC, another one is the RBC. WBC, which is responsible for the immunity and the related works. We will discuss in a zoology immunity topic. RBC is red blood cells, which is responsible for production of hemoglobin and oxygen carrying some things. Clear? So hemoglobin is synthesis in the RBC only. When the person suffering from sickle cell anemia, that carrier cannot synthesize hemoglobin. Cannot synthesis means that hemoglobin synthesis level will be reduced up to 50%. That is the first point. The second one is the shape of RBC. The shape of RBC will be changed. That shape of RBC will be, so the shape of RBC cell changed into sickle shape. That's why the disease is called as a sickle cell anemia. It becomes a normal RBC cell shape will be changed into sickle shape. That's why the disease is called as a sickle cell anemia. The third one is, we know very well that blood cells, hemoglobin, specifically hemoglobins are responsible for the oxygen carrier. That's why hemoglobins are called as a oxygen boat. This is an important Neet question mark, kindly note it. The hemoglobins are called as the oxygen boat, because that oxygens are carried by the hemoglobin of RBC cells. Here, that sickle cell, that RBC cell shape will be sickled one. It is not a regular in shape. So it cannot carry the oxygen level. In much quantity. So oxygen carrying capacity also reduced due to sickle shape. So these are three characters, three different characters. First one is hemoglobin level will be synthesized. The shell shape also changed into sickle shape. The third one is oxygen carrying capacity also reduced. The three characters will be three unrelated characters. It is not a unique character. Each one is an unrelated one. So the three unrelated characters are controlled by the single gene, HPS genes. This is an example for the pleiotropic one. This is an example for the introgenic interaction. The sickle cell anemia is an example for the introgenic interactions, which is also called as a intra-allelic interaction. Why it is called as introgenic? That interactions, reactions will be occurs within a gene. Reactions occurs from the within a genes, because in sixth amino acid, in sickle cell anemia, sixth amino acid, we will discuss in genetic disorder, this level. Okay, we will discuss detailedly about sickle cell anemia in genetic diseases. The next one is another one example is a single gene can able to control the another character. For example, in Pisum sativum plant, single gene can influence the seed coat, can determine the seed coat color, red spot in leaf, and flower color. So that is also example for the pleiotropic gene. Single gene responsible for determining the seed coat color, red spot in leaf, flower color. That's all about the pleiotropic genes. Next one is a dihybrid cross. Next one is a dihybrid cross. So I completed the monohybrid cross in 2 laws and the exception of Mendelism. Clear? The next one is a dihybrid cross. What's the meaning of di? Di means a di. Di means a 2. Hybrid means, yesterday I told, hybrid. Hybrid means a combination of 2 different things, which is known as a hybrid. Di means a 2. So we are going to cross 2 different characters of a plant, Pisum sativum plant, which is known as a dihybrid cross. So here in dihybrid cross, involving 2 characters of crossing, that is called as a dihybrid cross. Here he selected only the 2 characters. He selected only the 2 characters. In monohybrid cross, he selected only 1 character, which is known as a height. In dihybrid cross, he selected 2 characters. What are they? First one is a seed form. Seed form, which is also known as a seed shape. So here he selected, first one is a round, another one is a wrinkled. Seed shape is a 2 alternatives. What are they mean? So one is a round, another one is a wrinkled. Round is a dominant one, wrinkled is a recessive one. Clear? The next one is a Cotyledon color, which is also known as a seed color. Because cotyledons are produced from the seeds. So which is known as a seed color or a Cotyledon color. Here are 2 alternatives of there. One is a yellow, another one is a green. Another one is a green. Yellow is a dominant one, green is a recessive one. So the caps letter indicates the dominant one, small letter which is indicates the recessive one. Clear? Next, go for dihybrid crossing section. That gamete, the parent gametes are caps Y, caps R, and caps R. Which that gametes are responsible for producing the yellow round. That small letter Y, and a small r, which is responsible for making a green wrinkled. Or caps R is a round one, small r is a wrinkled one, caps Y is a yellow color, small y is responsible for producing the green color. Clear? So parental one is a caps Y, caps R. That is a dominant one. Small y, small r, which is a recessive parental one. So we are going to crossing the 2. So normally, you know very well that gametes undergoes a segregation process. After that only that gamete will be fused each other and forms a zygote, new zygote, clear. So similarly that the gametes are this homozygous only, that gametes will be segregated caps Y and small caps Y and caps R. Similarly, that green wrinkled gametes also segregated small y and small r. I wrote the dominant character in blue in color. I wrote recessive character in a green in color. So that gametes are crossing together. At that time, we can get it a caps Y, caps R, small r. In F1 generation, we are getting the yellow round plant. In F1 generation, we are getting the yellow round plant. So automatically, we know very well which undergoes a selfing, I mean self-pollination process. So automatically that each and every gametes will be, I mean that male gametes will be fused with the female gametes and forms a new gamete one, which is responsible for the production of a particular phenotypic character. For example, caps Y and caps R is first gamete. The next one, caps Y, small r. Next one, small y, caps R, small y, small r. That is one gamete. That each gamete will be interacted with each one. So at that time, the first one. That gamete will be, that male gamete will be fused with the female gamete one. At that time, that caps Y, we are getting the caps Y, caps R, caps R. Similarly, this gamete. Similarly, here that gametes will be crossed with that caps Y, caps R is crossed with the second gamete. At the time, we are getting the caps Y, caps R, small r. The next one, this third row, the caps Y, caps Y, caps R is interacted with the caps Y, small y, caps R, small r. And finally, which is crossed with the caps Y, small r is crossed with the small y, small r. So similarly, that each and every gametes, it will be crossed like this. At the time, we are getting the few gametes, so many gametes like that. So here we have to calculate it. Okay, the dihybrid is here. I explained by using the checkerboard method. So we must first we must identify the phenotypic character. How we identify the phenotypic character? First, we must write the dominant one. Dominant character, we should write it. First, dominant character is dominant is which one? Yellow is a dominant one. In color, yellow color is a dominant one. In shape, round is a dominant one. So we should write first dominant character is a yellow color. Second shape is a round in color. So first we should write like this. Second one is a color is a yellow. Two options is there. One is both are dominant one. Another one is both are dominant means yellow is a dominant. Color and shape is a dominant one. So yellow is a dominant, round is a shape, that is a dominant one. The second case is yellow is a dominant one, color is a dominant one. Another option is wrinkled, that is a recessive one. The third we should write like this. Third one is green. First we should write a color, then only shape, then green. Green is a recessive. In shape, round is a dominant one. So we should write green round. The last one is a green, missing one is a green wrinkled. The green wrinkled is the shape and color, both are a recessive one. So we should write like this. So we should count it. First, yellow round. How many yellow rounds are present? How many yellow rounds are present? Here I write in clean. Already I wrote in clean color. 1, 2, 3, 4, 5, 6, 7. So totally 9 yellow round is present. So yellow round means it should consist of the caps Y and caps R. Clear. The second one is yellow wrinkled. So totally 3 yellow wrinkled gametes will be there. The yellow green round is a 3. So yellow round is 9, yellow wrinkled is 3, green round is 3, green wrinkled is only 1, the last one, only 1. So we can calculate the phenotypic ratio. Phenotypic ratio means yesterday I told visible characters, morphological characters. That is called as a phenotypic ratio. So while we are considering that phenotypic ratio, at that time we cannot consider about the genes, genotypic one. Clear? So visible one. So totally 9 yellow round is present, 3 yellow wrinkled, 3 green round. One is a green wrinkled. So the phenotypic ratio is a 9:3:3:1. That is a phenotypic ratio of dihybrid cross. This is a most important NEET question, the frequently asking question. Already asked in the NEET. That is a NEET question. Phenotypic ratio of dihybrid cross 9:3:3:1. The next one is a genotypic ratio. We should know the genotypic ratio also. It may ask in NEET exams. So genotypic ratio means based on that homozygous and heterozygous, we can calculate the genotypic ratio. So first one is a homozygous yellow and homozygous round. Homozygous yellow. First we have to preference to the, we must give preference to the purity and color and shape. Just now I told. So homozygous yellow, homozygous round. How many? Which is not known as a caps Y, all are a caps letter. Caps Y, caps R is a only one. All homozygous is a only one plant is there. The next one, second one, homozygous yellow, heterozygous round. That is a 2 plant. Caps Y, caps R smaller. That is a 2 plant. The third one is a heterozygous yellow and homozygous round. Caps Y, small y, caps R. 2, we are getting we got a 2 plant. The fourth one is a heterozygous yellow and heterozygous round. Caps Y, small y, caps R, small r. We got a 4 plants. Next one, homozygous yellow and homozygous wrinkled. Caps Y, small r, small r. We got only 1 plant. Heterozygous yellow and homozygous wrinkled. Genotype is a caps Y, small y, caps R, small r. We got 2 plant. Next one is a homozygous green and homozygous round. Small y, caps R. Only 1 plant. Next one is a homozygous green and heterozygous round. Small y, caps R, small r. We got 2 plant. The last one is a homozygous green and homozygous wrinkled. We got only 1 plant. So we must write the which one is dominant one. The next one is which one is dominant color, homozygous. We have to write in order. So finally, recessive color, recessive shape, and similarly homozygous recessive color, homozygous recessive shape, like a homozygous green, homozygous wrinkle. First we have to write it. After that we have to find the genotype and we can write the genotypic ratio. So genotypic ratio is 1:2:2:4:1:1:2:1. That is the genotypic ratio of dihybrid cross. Again, I repeat, 1:2:4:1:2:1:1. That is the genotypic ratio of dihybrid cross. You can easily remember 1:22, 4:1:2, 1:1. So you can easily remember like this. Clear? So that's all about the dihybrid cross. Next one is a test dihybrid cross. Next one is the test dihybrid cross. So yesterday we discussed in monohybrid test cross and back cross. Here also similarly, instead of that mono hybrid, here we are using the dihybrid for test cross. Yesterday I told test cross means test cross means what? Good. Test cross means we are good. Test cross which is used to check the heterogeneity of gametes. Test cross which is used to check the heterogeneity of gametes. Clear? Test cross means we are getting the one progeny in F1 generation, which is crossed with the recessive parent of dihybrid, which is crossed with the recessive character, recessive parental one. So we are going to cross the F1 generation progeny. F1 generation progeny is crossed with recessive parental in dihybrid, which is called as a test cross of dihybrid. As usual, process will be started. First one is a gamete will be segregated. Gametes will be segregated. Caps Y, caps R, caps Y, small r, caps R, small y, small r. And the last in recessive characters, similarly caps R and small r and small y. I wrote in green in color. Recessive characters I wrote in a green in color. Clear? So small r and small y, that gamete is segregated from the recessive parental one. Here, that F1 progeny gametes, segregated gametes will be crossed with the recessive parental one. So at that time we are getting the 4 gametes. What are the means? Caps 1, first one. This caps Y, caps are crossed with caps R small y, small r and caps small y. At the time we are getting the caps Y, small y, caps R, small r. Similarly, each and every gametes will be crossed with small r and small y. So at that time we are getting the 4 kinds of gametes. Like end of the result is we are getting the 4 kinds of gametes. What are they means? Caps Y, caps R, small r, caps Y, small r, caps R, small y. The last gamete is a small y, and small r. So finally we are getting the 4 plants. What are they means? Yellow round and yellow wrinkled and green round and green wrinkled. Clear? So end of the test cross of dihybrid, we are getting the 4 kind of plants. What are they means? Yellow round, yellow wrinkled, green round and green wrinkled. So that phenotypic ratio and genotypic ratio both are same. I mean phenotypic ratio is 1:1:1:1, all 1. Genotypic ratio also 1:1:1:1, all 1. So here the phenotypic ratio and the genotypic ratio are same in test cross of dihybrid. Clear? That's all about the dihybrid. Then next Mendel's third law. Third law is, you know very well, law of independent assortment. Law of independent assortment. This law is purely based on this law is formulated purely based on the dihybrid cross. The first 2 law, yesterday I told the first 2 law is formulated based on the monohybrid cross. That last third law, law of independent assortment, is formulated based on the dihybrid cross. Here, independent assortment. Independent, what is the meaning of independent? Independent means does not depending anything. That is the meaning of independent. Freedom, independent means freedom, does not depend anything. That is called as independent. Here, the genes for each pair of characters will separate independently. Separate independently from those of other characters during gamete formation. The genes for each pair of characters separate independently from those of other characters during gamete formation. Listen, ma. Listen here that one factors caps y, caps r, small r. That is a factors. Nowadays we know very well that is a genes. It will be separated, segregated like caps y, caps r, or small y, caps y, small r, caps r. So, small y, small r, like this. It will be segregated like this. During that segregation time, one won't interact with other one. It will be segregated independently. For example, caps y, caps r will be separate independently. Does not interfere with the next gamete. Caps y, small caps y, small r does not interfere with the caps y and small r. Clear. So during that gamete separation, it will be separate independently, does not depend on others, does not interfere with others. That is called as a law of independent assortment. The next one is a chromosomal theory of inheritance. Next one is a chromosomal theory of inheritance. The chromosomal theory of inheritance very first proposed by the W.S. Shudden and Bovary in the year of 1902. That question may ask in Neet, ma. So kindly note it. The chromosomal theory of inheritance, which is proposed by W.S. Shudden and Bovary in the year of 1902. So according to this theory, gene located on the specific location of chromosomes. The gene will be located in the specific location of chromosomes. That is known as a gene locus. Location. What's the meaning of locus? Location. Gene will be present in the specific place of the chromosome. That is called as a gene loci. It is called as a gene loci. Here also the gene will be present in the particular place of chromosome. This chromosomal theory states that chromosomes are a vehicle of heredity information and expression as genes are present over them. So yesterday I told chromosomes are a vehicle. Because that the gene will be present in the chromosome. So genes are responsible for all. Genes can able to carry the hereditary informations. That's why I'm saying that chromosomes are a vehicle of a hereditary information. That theory states that thing only. That chromosomal theory, while we are comparing that Mendelian statement with the chromosomal theory, that both will be behave parallel. That's why I wrote here that parallel behavior of Mendelian factor. Parallel behavior of Mendelian factor. So factor means caps T, small t is called as a factor. Caps y, caps r. As a factor. Clear. So in Mendel used the term factor. Nowadays that factor is known as a gene. Clear. So the parallel behavior of Mendelian factor and the chromosome, it will be behave like a parallel when during the meiosis time and fertilization time. During the meiosis and fertilization. So meiosis means we know very well that gamete formation. Meiosis means known as a gamete formation. Meiosis means reduction stage. Diploids becomes a haploid. That is called as a meiosis. So meiosis, gamete will be formed. And fertilization. So fertilization means that the formed gametes will be fused together and forms a diploid stage. That two haploid cells will be formed. The two haploid gametes will be formed and forms a diploid one. That is called as a fertilization. So during that meiosis and fertilization time, that Mendelian factors and chromosomal theory, that both will be like a parallel, behaves like a parallel one. So that thing is I explain in tabular column. So parallel behavior of factors, I mean gene and chromosome. The first one is the chromosomal theory. The both are state the same meaning. I'm saying that finally I'm trying to say that thing only, that both are saying the state the same things only. For example, first one is that chromosomes occurs in homologous pair. Chromosomes occurs in homologous pair like this. It's look like a homologous, similar things. Similarly, that gene in, I mean factors, which is also occurs in allergic in pair. Allergic in pair. For example, you can see that T is present in one allele because allele means alternative forms. Caps T present in one loci of chromosome, small t is present in the another loci of chromosome. So that is also like a homologous. Clear? So chromosomes occurs in a homologous pair. Similarly, the genes also occurs in allergic pair. The second point, during that gamete formation in chromosomes, during that gamete formation, that gamete will be segregated. The gamete will be segregated, and which is enter into the gamete. So during that entry time, only one chromosome will be entered. Listen, ma. So during that gamete formation in chromosomal theory, so during that gamete formation, so normally the gamete will be separated. Chromosomes will be separated. The separated chromosomes, I mean the diploid stage, chromosomes will be separated. For example, in human sex chromosome, in male consists of the XX chromosomes, in female consists of the XX chromosome, male consists of the heterogenic XY chromosomes. So that is a diploid in nature. So while the new gamete is formation, at that time, in case if you are taking a male means X chromosomes will be separated. Similarly, Y chromosomes will be separated independently. That separated X chromosomes will be enter into the new gamete. For example, X may be combined with the another one X. Otherwise, X may be combined with the Y chromosome, like that. Clear? So, in chromosomal theory, the chromosomes will be, the diploid stage of chromosomes will be separate independently. That separated chromosomes will be entered into the new gamete during the gamete formation. That separation occurs independently. During that separation time, one chromosome does not affect the another chromosome separation. Clear? Similarly, factors, gene also separates during that gamete formations. Normally, we know very well in monohybrid cross, does the dihybrid cross, all things. For example, Mendel's second law, law of segregation. That thing only here I explained. So during that gamete formation, that both gametes in a factor, that gametes will be separated like this. For example, small T, small t is present. That is a factor. So during that gamete formation, it will be separated. Gametes are separated like a caps T and a small t. After that only the new gametes will be formed by a fusion of two gametes. Clear? So this is that Mendelian factors also, the Mendelian also follows the chromosomal theory. Clear? So that's why I'm saying that that chromosomal theory and Mendelian inheritance both are behaves like a parallel one. That's all about the chromosomal theory of inheritance. Next one is a cytoplasmic inheritance. What's the meaning of cytoplasmic inheritance means? Cytoplasmic. What's the meaning of cytoplasm? Cytoplasm means you know very well. What's the meaning of protoplasm? Then only you can say the answer for cytoplasm. Protoplasm means water plus some inorganic molecules. Water plus some inorganic molecules, proteins, lipids, and RNA like that. Water plus inorganic molecules are present it. So where the protoplasm is present, based on that place, it will be named as cytoplasm or maybe a nucleoplasm or etc. etc. Clear? So that inside that cell, cytoplasm will be present it inside the cell. I mean that inside the plasma membrane, all organelles will be floating in the cytoplasm. All organelles are floating in the cytoplasm. Clear? All organelles are floating in the cytoplasm. Inside that one, that nucleus will be present it. That nuclear membrane is separated the nucleus from the cytoplasm. Nuclear membrane is separated the nucleus from the cytoplasm. That's why in case if the protoplasm is present inside the nucleus, which is called as a nucleoplasm. Clear? So cytoplasm means we know very well, water plus some molecules. This is called as a cytoplasm. Here the cytoplasm, many organelles will be present it. That organelles, cytoplasm means you know very well. What's the meaning of inheritance? Inheritance means a transfer of character from one to next generation. Transfer of character from one to next generation. That is called as a inheritance. Here the particular character will be inherited from one to next by cytoplasmic organelles. By cytoplasmic organelles. Organelles present in the cytoplasm. That is called as a cytoplasmic inheritance. In cytoplasmic inheritance, we will discuss detailedly now. The cytoplasmic inheritance, also known as a extra chromosomal inheritance. Extra chromosomal inheritance, which is also known as a extra nuclear inheritance. Why which is called as a extra chromosomal or extra nuclear inheritance means that chromosomes, normally we know very well that DNA will be present inside the nucleus. DNA will be, I mean DNA gene will be present in the chromosomes. Chromosomes are present in the nucleus. So we know very well that DNA will be present in the nucleus only. So apart from nucleus, DNA will be present in another two organelles also, namely mitochondria and chloroplast. Namely mitochondria and chloroplast. That mitochondria and chloroplasts are present in the cytoplasm of cell. Clear? That mitochondria and chloroplast having the DNA which is present in the cytoplasm. Clear? So that's why which is called as a extra chromosomal inheritance. So already the chromosomes are present inside the nucleus one, apart from nucleus, which is present in mitochondria and chloroplast. That's why which is called as a extra chromosomal inheritance and extra nuclear inheritance. That mitochondrial DNA or a chloroplast DNA, it can able to capacity to transfer the character from one to next one. So that's why which is cytoplasmic inheritance also called as a extra chromosomal or a extra nuclear inheritance. That cytoplasmic inheritance also known as inheritance, also known as a non-Mendelian inheritance. Why which is called as a non-Mendelian inheritance? You know very well, Mendel states that DNA can only transmit the character from one to next one. Clear? Which is present in the nucleus. That is the Mendelian statement. Clear? But in cytoplasmic inheritance, apart from that nuclear DNA, other DNAs of chloroplast and mitochondria can able to inherit the character from one to next generation. That's why that cytoplasmic inheritance also known as a non-Mendelian inheritance. Clear? So in cytoplasmic inheritance, plasma genes, which is important one. There is a neat question. Plasma genes, that is a neat question. What's the meaning of plasma genes? Plasma genes are nothing. It is an independent, self-replicating hereditary material. Plasma genes are independent, self-replicating hereditary materials will be present in the cytoplasmic organelles of mitochondria and chloroplast, which is known as a plasma genes. Again, I repeat, which is important one. It is a neat question. Independent self-replicating genes or factors which is present in the cytoplasmic organelles or mitochondria or chloroplast, which is called as a plasmagens. That plasmagens are a extra chromosomes. That plasmagens will be act as a extra chromosomes. So here in cytoplasmic inheritance, two kind of inheritance will be available. One is a mitochondrial inheritance. Second one is a chloroplast inheritance. So I will explain the cytoplasmic inheritance with the help of a chloroplast inheritance. Here in 4 o'clock plant. 4 o'clock plant means Miraphilus jalapa. Miraphilus jalapa, which is known as a 4 o'clock plant. So normally that Miraphilus jalapa shows a variegation in their leaves. Normally that 4 o'clock plant shows some variegation in their leaves. Why that variegation occurs means presence of or absence of chlorophyll in the chloroplast. So the 4 o'clock plant shows different colors of leaves. Why? The chlorophyll may be present or maybe chloroplast may be absent in the particular place. Clear? So that's why it shows us some variegation on the leaves of 4 o'clock plant. Clear? That thing will be controlled by the chloroplast DNA or chloroplast DNA. Clear? In case if you are taking that 4 o'clock plant, so many plants are look like. So many plants are, for example, some crotons are shows so many different colors of leaves. Clear? So in 4 o'clock plant, if you are taking one leaf means it is a dark in color, which is a dark in color. There that chlorophyll will be present rich. Clear? Similarly, if you are taking another one leaf, the same plant, the same 4 o'clock plant, which shows a pale green color. Pale green color means their chloroplast content is too low. Because why means it may occurs the 3 kinds of plastids will be there. You know very well, leucoplast like this looking. So why that pale green color occurs means less quantity of chlorophyll may be present. So at the time also it's look like a pale green color. Clear? So that thing will be experimentally proven with the help of the same 4 o'clock plant. The first one, he took pollen grains of pale green color leaf of 4 o'clock plant. Why I noted a male means we are taking a pollen grains. We are taking a pollen grains. That's why which is denoted a male. Otherwise, which is introduced into the another plant flower of stigma of plant. That's why which is I denoted the female one. Clear? So the first one, the pollen grains, which is transferred to the female stigma of another plant. That another plant is a dark green color. We are taking a pollen grains from the pale green color leaf of plant, which is that pollen grains are introduced into the stigma of dark green color parental plant. So end of the result, we are getting the, so while we are crossing the pale green color leaves, which is crossing with the dark green color one, at that time we are getting we got a dark green color leaf. Clear? Similarly, dark green color male. The reverse one, the dark green color male is crossed with the pale green color female. At that time also we got a pale green color leaf. So why that experiment shows, why the female character only expressing? In first experiment, dark green color, that is a character of female plant. In second experiment, pale green color will be expressing. Here also that pale green color will be expressing. Why that female character only expressing? Because already I told there is a cytoplasmic inheritance. Because of chloroplast only provides the ovum. Ovum will be present in the female plant. Ovum will be present in the female one. Ovum only provides the 100% cytoplasm. During the fertilization time, ovum will be produce the 100% of cytoplasm. That male provides only the nucleus. The male provides only the nucleus. Cytoplasms are provided by the ovum only. So here, this is a cytoplasmic ingredients only. So ovum will be that 100% of ovum will be provided by the female plant only. That's why that female character will be expressed. That cytoplasm, that chloroplast where it is present. Just now I told cytoplasm of the cells, cytoplasm of the cell. Clear? Cytoplasm of the cell. That's why which is example for here that nucleus is not transferring. That nucleus is not determine the green color. Here that chlorophyll of chloroplast only determine the particular character. Clear? So that's why which is example for the cytoplasmic inheritance. So now time is over. So next topics, linkages and other things, linkages, sex determinations, gene interactions, everything we will discuss in tomorrow. So today, what are the topics we are discussing, ma? Anyone? So recalling is a most important one. Today we discussed about the exception of a Mendelian. So the four things are important: incomplete dominance, codominance, multiple allele, pleiotrophic gene. Next one is we discussed about dihybrid cross. We discussed about the dihybrid cross. Next one is we discussed about the chromosomal theory of inheritance, test cross of dihybrid, law of independent assortment. The last one we discussed about a cytoplasmic inheritance. You must recall it. Clear? Thank you. Today class is over for. Thank you.